Okay, so hello everyone. Welcome to this time's Immune Zoom seminar. So again, we have a treat today. It's really my great pleasure to welcome today's speaker, Dr. Eran Elinav. So I think a lot of us already know Eran's work, but just a brief introduction. Eran is currently a professor heading the Department of System Immunology in Weizmann Institute of Science in Israel. And he also is the director of the Microbiome and Cancer Division, National German Cancer Center in Heidelberg. So his lab at the Weizmann Institute and in Heidelberg are mainly focused on studying the molecular basis of host microbiome interactions and their effects on health and disease, with a goal of personalizing medicine and nutrition. Actually, Eran has a pretty nice book from 2017, Personalized Diet. If you guys are interested, I suggest you check it out. I think it's pretty interesting. So Eran actually completed his MD degree at Hebrew University in Israel, and then followed by a clinical internship, a residency in internal medicine and physician scientist position at the Tel Aviv Medical Center Gastrology Institute. And then he received a PhD in immunology from the Weizmann Institute in Israel, followed by a postdoc training in Yale with Dr. Richard Flawell. So Eran has really very productive both in his PhD study and also postdoc study, but particular after he started his independent study group, he really actually expanded his research horizon to different area. And he published really abundantly more than 220 publications, including major recent discoveries related to the effect of host genetics, innate immune function, and the environment factors such as dietary composition and timing. I think a lot of us actually, and personally, at least myself, really impressed with his pioneer work in spatial and time dimension regulation with the microbiome and host interaction. Based on these work, he really received numerous honors and awards, including the Clare and Emerald Award from the American Physicians of Medicine, the Eran Foundation Awards, the Levinson Award for Basic Science Research, and he is also currently a guest professor in University of Science and Technology in China, and he's elected member of European Molecular Biology Organization, and also elected fellow in American Academy of Microbiology, and then international scholar at the Howard Hughes Medical Institution and the Bill Melinda Gates Foundation. So I think his topic for today is he's going to talk about host microbiome interactions in health and disease. And then without further ado, please Eran, we are looking forward to your seminar.
Thanks for this overly kind introduction, and I'm happy to be with you by Zoom. And as you heard, my group and I extensively study host microbiome interactions and their potential impacts on human physiology and human disease. You know, they say a photo is worth 1,000 words. This may be one of them. An electron microscopy photo taken at a magnification of 160,000x from a healthy terminal ileum, and you can see the bumps representing the villi, and above them you see what looks like a carpet of shoelaces. This is your healthy microbiome. You can also already appreciate how intimate the interaction is between these trillions of microbes of thousands of different families and our human part of the holobiome. And to start my talk, I decided to give you my 2 cents on 4 major concepts which have emerged during the first decade of microbiome research. This is a super young field, and these will take us towards the rest of the presentation. So the first hallmark concept relates to the fact that we mostly study the gut microbiome because it's the most easily accessible and the richest ecosystem in our body. But we need to remember that the microbiome exists at every site at our body that interacts with the outside world, including the oral cavity, the respiratory tract, the skin, the genitourinary tract, and even within tumors, there are low biomass microbial populations. And we extensively and increasingly look at these interesting niches and the contributions of the microbes in these niches into their function. The second hallmark relates to the fact that if I were to sequence all of your microbiomes deeply enough or extensively enough, I would find that each of us carries a unique signature or fingerprint of a microbiome. This was very, very scary to us a decade ago when we started together with the field to explore the microbiome, but we've learned to harness this inter-individual variability towards trying to explain how the microbiome may lead to different clinical outcomes in people featuring the same genetic risk. And I'll show you at least one example from our own work. A third concept that we follow relates to the microbiome as a signaling hub that integrates into it many signals that may come from within the human body. For example, the human genetic or immune system impact the composition and the function of the microbiome, but also a large variety of environmental factors, such as the ones exemplified in this cartoon, impact our microbiome. And all of these signals are perceived by these ecosystems, processed and transmitted to the host in a fascinating communication network in which, and we see our mission in life as decoding one letter at a time, the alphabet, which forms this inter-kingdom communication network, and what goes wrong when it is perturbed that leads to a risk of developing disease. And finally, the last hallmark, which has been developed in the last 5 years of microbiome research, relates to the fact that in addition to local effects mediated by the microbes, they are also regarded as a biochemical factory that generates or modulates thousands of potentially bioactive small molecules, which we call metabolites. And we increasingly realize that these small molecules could influx into the sterile host and reach every cell and organ in our body. And this can start explaining us how microbiomes in one place of the body could impact processes that occur miles away. And so this is a very complicated set of ecosystems, and their study has been facilitated by several technological breakthroughs, including shotgun metagenomic sequencing that enables us to sequence each gene in each bug in a microbiome community to start understanding their composition and functional potential. Mass spec-based metabolomics, in which we characterize these thousands of microbiome-associated small molecules. Culturomics, which are very extensive, high-throughput systems, which we and others have developed in the last 2 years, which finally enable us to sequence, to culture many of the previously unculturable bugs. And in a vitro or ex vivo system that enable us to model the microbiome, for example, organoids and organ-on-chips. But still, I think the major workhorse in transforming from observation and correlation into causation are germ-free mice, these sterile mice that are housed in the specialized isolators. You see here the very large Weizmann Institute germ-free facility. And these enable us to model human or animal disease and the contribution of the microbiome to this disease by transferring the microbiome into recipient mice and modeling disease process in these recipients. And I'll show you 1 or 2 examples from our own work. And by using all of these technologies, we see or we hope that our potential contribution would be to try and enable better mechanistic understanding of host microbiome interactions, including how the microbiome impact human physiology and health, how the human body may impact the composition and the function of the microbiome, and what happens when the communication channels between the 2 partners of the holobiont are perturbed that lead to a risk of developing many multifactorial diseases. And by doing so, the field is slowly able to show that we may be able to reach new principles facilitating the development of microbiome-mediated disease treatment. And what I chose to share with you today are my 2 cents on where I see our place in terms of microbiome-associated disease treatment. And to be honest, in 2022, I think that microbiome-associated treatment is mainly composed of 3 relatively non-specific and in many cases not sufficiently evidence-based avenues of treatment, which include prebiotic therapy, which is this fiber-based, non-specific dietary intervention aiming to make the microbiome healthier, whatever that means, I don't know. Fecal microbiome transplantation, which has been very successful in some contexts, and this includes the transfer of a sick person, of a healthy person's microbiome into a sick person, and this has been shown to be extremely effective in diseases such as Clostridium difficile colitis, the most common hospital-acquired infection in the developed world, and probiotics, which were there before we knew there was a microbiome, and I'm not going to go into probiotics today, a rather painful subject. But what's more encouraging is the fact that with the explosion of microbiome research, we're slowly able to integrate this data into new experimental modes of rational and maybe more science and data-driven microbiome interventions. And what I'll do in the next few minutes, I chose to share with you 5 of these avenues that we follow in terms of developing new modalities of microbiome-related treatment. And the first modality is our data-driven version of the prebiotic therapy, which we term personalized nutrition. So over almost a decade ago, when we started, we already realized that by far nutrition is the biggest environmental modulator of the composition and the function of the microbiome. But when I started looking into all the systems that we intuitively use in our daily lives, when we try to come up with what we hope would be a healthy diet for ourselves, I realized that most of this is based on very small scale studies in the 1970s, which gave different food or food components a value or a number. And based on these numbers, we all try to intuitively mix and match diets in order to make them better for ourselves. For example, calories are such a grading system, which gives numbers to different food components. An even more widely used system that is at the basis of any diet that you've ever tried, whether you went to a dietitian, a physician, or even if you bought a book at the airport, is called the glycemic index. And this is a system that's based on very small scale human trials that were conducted in the 1970s and usually included 10 healthy individuals who were given an identical food, and then their blood sugar levels were measured for 2 hours. And so if you were to take 10 healthy individuals, give them an identical piece of celery, you would find that the blood sugar level in average spikes, just like the black graph. You would be given this spike a number, and this would be denoted as the glycemic index of celery. Now, if you were to ask the same healthy 10 individuals to kindly eat an identical piece of chocolate cake, their average rise in blood sugar levels would look something like the blue graph. It would be given a higher number, and this number would be denoted as the glycemic index of chocolate cake. Now, if you look in your smartphone, you would find endless tables that give glycemic index values to any food or food component on Earth. And again, take my word for it, almost any diet that you've ever thought about is based in one way or another on this glycemic index criteria. Now, here comes the big problem. The big problem was that when we tried this method from the 1970s, but on a larger cohort of individuals, it just didn't work. And this started with our first observation in 2014, in which we studied the contribution of non-nutritive sweeteners on the microbiome and on metabolic health. And as part of these mouse-based studies, we performed a small-scale human trial in which we gave a group of individuals an identical amount of saccharin as an example of one of these sweeteners. And to our surprise, half the people reacted in a significant spike of blood sugar levels, the other half didn't care, meaning that already inherently, this grading system for the food or for the sweetener in this case that gives a number to that sweetener is inherently flawed because some people react opposite than the others. And this made us enlarge the effort and collect 1,000 people in which we extensively measured their glycemic responses to test food or to real life foods. And again, the average responses of these 1,000 people to any test food we've given, even for 50 grams of glucose, was exactly the glycemic index of that food. But when we looked at the individualized levels, the variability was unimaginably high. So some people eating 50 grams of glucose spiked to the roof, others didn't care at all. So already intuitively, this notion of a one-size-fits-all diet, which we've all been we've been chasing for 50 years or more cannot be true, because if your glycemic response to given food is opposite than mine, then the same food cannot be good for both of us. So this eureka moment was the basis of a very ambitious project, which we termed the Personalized Nutrition Project, in which we took this 1,000 people cohort, and we kindly asked these people to give us a week of their life while collecting at the time an unprecedented amount of data, including of course the gut microbiome, which is what we do in live, blood tests, questionnaires. We developed a smartphone app for this project, so they gave us a real life point of care kind of assessment of what they were doing during a week of follow-up, and we connected them to a continuous glucose monitor, which measures blood sugar levels every 5 minutes for an entire week. So rather than having 10 individuals with 6 skin pricks from the 1970s, here we had 1,000 people, each giving us 2,500 accurate glucose measurements. And taking all of this data from each of the individuals, our students used machine learning and AI technologies to train a predictive algorithm that would enable to predict a person's response to any given food, including foods they've never consumed. And so the next slide is a little bit unfair because it summarizes 7 years of work in one slide, but it tells you on the left what the gold standard is of our predictability of a dietary response. When we go to a physician and ask to be given a diet that would lower blood sugar levels, there's some merit to it because we are usually given a diet very low in carbohydrates, but the predictability is extremely low, 0.38. When we use our data-driven or science-driven predictive approach, we dramatically increase our ability to predict a person's response to any given diet. And as you do in these drills, at some point you need to stop the training of the algorithm and apply the frozen algorithm on a fresh cohort, in this case 100 people, and we were quite pleased to see that the predictability was maintained. And of course, this made us pleased, but it was also time to put ourselves to the real life test, which was done in many different contexts. For the sake of time, I'm just showing you one, which is a recently published large randomized controlled trial in which we took 200 pre-diabetic individuals. These are individuals already suffering from disturbances in blood sugar control. It is estimated, for example, that 40% of the adult US population are pre-diabetic, so we have very little to offer them. We randomized them into the best ever diet prescribed, which is a Mediterranean low carbohydrate diet. And the other half got this data-driven algorithmic diet, which in many cases is counterintuitive. A person could have some ice cream, which is good for him, some tomatoes that are bad. You never know until you measure them. We followed them for a whole year. And just to give you an example of the many results in this study, you can see in red 3 different glycemic indices over the year of follow-up, in this case over the year of follow-up, that show that the standard of care diet does have a merit. It mildly improves glycemic control. This has been shown for decades. But in contrast, in green, you can see that the personalized diet outperforms the standard of care diet by quite a lot, and efficiency, durability, and compliance are dramatically improved with this dietary approach. And just to show you that we don't stop there, many people have now validated this in other cohorts, including from North America. But we think that by measuring, we may be able to harness that into even non-metabolic contexts. So we have a very large international effort to characterize the effects of diet on inflammation. There are other efforts that we're engaged with respect to cancer, and I think this is just the beginning of this more measurement-based field. So this was an example of how rationally integrating diet and the effect of diet on the microbiome, because what I didn't tell you for the sake of time is that many of the features that the machine learning algorithms use are microbiome-based, and we can actually just use the microbiome plus some clinical features to get at a better predictability today when we did over 100,000 people using this approach. But this is the dietary approach that we are trying to develop. The second avenue of microbiome-associated research relates to the transfer of live microbes. So I've mentioned probiotics, I've mentioned fecal microbiome transplantation, but I want to kind of give you a vignette of, I think, an interesting study that we've published a couple of years ago related to one of these other non-gut microbiomes, which is the vaginal microbiome. And this is a very interesting microbiome. It's much simpler than the gut. It's dominated by lactobacilli, secreting many factors, including acid that are important for this niche. But in a very common disease, actually the most common infectious disease that I know of, which is called bacterial vaginosis, which impacts between 1/3 and 1/2 of all women worldwide, this lactobacilli-dominated microbiome is replaced by an anaerobic configuration, which leads to many symptoms related to discharge, smell, interrelational problems, and so on and so forth. Now, most women take antibiotics and get better, but a rather large subset or minority of these affected women, around 10% of all women worldwide, you can make up the numbers, develop a form of antibiotic-resistant bacterial vaginosis, which we call intractable or recurrent bacterial vaginosis, which no longer responds to our treatment. So this is a huge unmet need. And in addition to the symptoms I've mentioned before, these women with a recurrent or intractable BV develop a dramatic risk to suffer from a number of obstetric and gynecological complications, including the ones depicted here. So again, this is a huge unmet need. And we ask a very naive question, which is, if fecal microbiome transplantation has been shown to be so effective in a dominant gut infection such as C. difficile colitis, maybe vaginal microbiome transplantation could do something to reset the vaginal, the bacterial vaginosis microbiome towards its healthy configuration. So we collaborated with a group of OBGYN experts in Israel. For a few years, we've calibrated the vaginal transplantation to be effective and safe. And when we felt that we were ready, we performed our first in-human clinical trial in a group of women suffering from intractable bacterial vaginosis and followed for decades without any solution. You can see in this graph the clinical score called AMSL criteria on the y-axis, and you can see that all of these women spike to the roof every few days. So this is really a terrible disease for these women. But after a successful vaginal microbiome transplantation for the first time in their life, we were able to generate complete remission in all of these women, which are now followed for over 4 years. And what was more interesting to us in this pilot trial was what we saw when we sequenced their vaginal microbiome. Now, this graph is called a principal component analysis graph. It enables dimensionality reduction of the microbiome. So each dot here represents one vaginal microbiome sample. And if two samples are very close to one another, the two microbiome configurations are very similar to one another. If two dots are far apart, the microbiomes are very different. So if you see the three full colored microbiomes that cluster together, these are the healthy donor microbiomes. So it means that the healthy vaginal microbiome is very similar to one another. The full red dots, which are clustered quite far away, are the intractable bacterial vaginosis samples. And this is all expected. But what's very interesting is the change in the configuration of the bacterial vaginosis sample. These are the arrows leading to the new place with these empty red configurations, which now clusters very similarly to the healthy vaginal microbiome after a successful vaginal microbiome transplantation, meaning that we were not only able to clinically improve these women, but we were able to reset and stably do so their vaginal microbiome configuration towards the healthy donor configuration. And this is now being followed by much larger controlled randomized trials, including in susceptible populations across the world, to try and learn about how to refine and how to upscale this new method and make it available to the many women who would potentially benefit from it. A third microbiome-associated treatment, which is making us and the field very excited, is one which we call postbiotic therapy. So we've talked about prebiotics, about probiotics, and now I'm talking about postbiotics. And this is a therapy related to the thousands of small molecules that I've mentioned at the beginning of my talk, these metabolites, that may be potentially bioactive. And if we understand what is missing in terms of these small molecules in a given disease context, or what is there in overly abundant, we can intervene through these small molecules by supplementing them or by blocking their signaling without ever having to deal with the microbes that make them, that are so diverse between people and make our lives so miserable. So this is what we call postbiotic therapy. And just to give you one vignette of one study of our own group, we had a very talented postdoc called Eran who is a neurobiologist, and he was very interested in exploring the potential microbiome gut-brain axis. And he chose to focus on a devastating neurodegenerative disease called ALS, or amyotrophic lateral sclerosis, a horrible disease impacting adult individuals and ultimately leading to death in 100% of them within 5 years of diagnosis. And this is a disease characterized by progressive muscle weakness. It's very variable in its clinical presentations, and this made us think that maybe there is some environmental modulatory activity that may or may not be related to the microbiome. So what was done in this trial, first in mice, in a genetic model of the familial ALS mutated gene, the SOD1 transgenic model, Eran followed these mice, and he used many different objective criteria related to progressive muscle weakness. You can see here 2 of these readouts. And what he saw, surprisingly, and if you focus on the blue graph, was that when he took these genetically ALS-prone mice and followed them over time, expectedly they develop a progressive exacerbation of disease manifesting as progressive weakness. But when he took the same exact mice and depleted their microbiome by administering wide-spectrum antibiotics or doing this in the germ-free environment, this is the red group, disease became much more exacerbated and much more rapid in our mouse facility, meaning that maybe something in the microbiome was modulating disease towards a more severe phenotype in our facility. So this, as you may imagine, led to a very extensive microbiome probing in trying to get some suspects that are associated with this worsened disease phenotype in our facility. We identified 11 different strains, and we focused on one of the strains, the only one that was associated with potentially a better outcome because it was missing as disease was progressing in these mice, and it was depleted by antibiotics. And this was a bug called Akkermansia municipula. And since we pride ourselves on trying to contribute towards causality, the next set of experiments I'm showing you are ones in which we supplemented this missing bug back to its physiological levels. And in this case, the red group is the control SOD1 transgenic ALS-prone group. And you can see again, in the 2 readouts I'm showing you, there are many more that I'm not showing you, how they develop this progressive disease a long time. But the black group, these are the identical mice that are now supplemented with this potentially good bug, develop a slower disease deterioration and a better outcome, meaning that somehow this strain of commensals in our facility was changing disease course towards a better presentation. And of course, the million-dollar question is, what is the molecular mechanism? So we've performed many experiments in which we tried to look into the different machineries this bug presents and the different small molecules that it modulates. We came up with a short list of 27 molecule suspects, and we ended up focusing on one nicotinamide, which seems to be missing during the disease process. And this is a molecule that is strongly secreted by this bug. And when it was missing, the molecule was missing, and so on and so forth. To prove causality, again, we supplemented this missing molecule back to its physiological level, this time systemically in ALS-prone mice. And you can see an example of one of the experiments in which the control groups in red developed the disease deterioration over time, while the nicotinamide supplemented group in green developed a slower disease, suggesting that at least one of the mechanisms is mediated by nicotinamide. And we've progressed further to show or to suggest what it may be doing neuronically in the motor neuron centers in the brain. I'm not showing you this for the sake of time. But what I am showing you is that in this study, we tried to scrape the surface and see whether maybe some of the conceptual findings that we found may be true in some humans with ALS. We've allocated a cohort of human ALS patients in Israel and their household controls. This is important because this at least partially controls for dietary variabilities. We sequenced their microbiomes, we characterized their metabolomic profiles, and we were able to show that the microbiome in ALS patients is quite distinct from healthy household controls, even during early stages of disease. And same we could show for the small molecule profiles. And interestingly, one of the differentially expressed molecules was the exact nicotinamide that I've shown you in mice, which was suppressed both systemically and in the CSF of these ALS patients as compared to household controls. And again, there's a lot of effort by many groups now to understand these potential connections mediated by nicotinamide or many other differentially expressed molecules, and maybe to intervene through some of the modalities I'm sharing with you in slowing down or doing something to this devastating cureless disease. Fourth avenue of treatment is that cited about unmet need in the field. So imagine what happens if you find a commensal strain that is strongly associated with the development of a disease or with whatever complication of disease you're interested in. How do you get rid of this commensal without impacting the entire ecosystem that I'm showing you is so important around it? And this is really an unmet need because antibiotics are a very bad way to do so. Antibiotics induce the emergence of resistant strains. They have many adverse effects. You cannot give them forever. And they're of course very non-specific. So we really have no way of doing it. And many people are trying many different things. What we are trying is to use bacteriophages, which are these viruses, which are unique because they specifically target bacteria. And we don't know of eukaryotic receptors for phages, so they're considered to be generally safe in humans. The problem is that bacteria and phages experience a huge arms race. You know, many of the Nobel Prizes in the last 50 years, including the discovery of restriction enzymes, CRISPR, and many more, have been shown to be related to this arms race. So a phage attacks a bacteria, the bacteria develops a resistant mechanism. And if you were to just use one phage against a bacteria of interest, there's a 100% chance of resistance developing very rapidly. So what we chose to try and do is to develop an approach in which we would generate a cocktail of phages, each distinctly attacking a bacteria of interest through different mechanisms and different receptors. And together, this cocktail would do the trick and would not enable the bacteria to develop resistance. This resulted in a very big project. The lab has been turned into a factory of phages, which are manufactured in sewer and in sinks of dentists and so on and so forth. And just to give you a short vignette, our most advanced project within this scope relates to IBD, to inflammatory bowel disease. And we've collected a rather large cohort from 4 different geographical locations, and we were able to deeply sequence and characterize a family of strains of a pathobiome that are associated quite strongly with disease exacerbation, and then developed a 5-phage cocktail against these this clade of pathobionts of disease-associated bacteria, test it extensively in vitro and show, and in vivo in preclinical models, and show that the eradication of these disease-associated commensal was associated with suppression of inflammatory responses in these preclinical models. And we were even able to bring this back to the bedside and basically both in a gut-like system and in a phase 1 trial show that members of this phage cocktail could be safely absorbed, could reach very high concentrations within the lower gastrointestinal tract that are expected to eradicate the bacteria in the right disease-associated population. So this gives you an example of one of the efforts underway to kind of generate a more silver bullet intervention that would kill a member of the microbiome when it's associated with disease without harming the entire ecosystem. And finally, I don't know how I'm doing in terms of time. I think I'll manage. And finally, the last avenue of microbiome-associated treatment I wanted to share with you is one which many of us tend to forget, which relates to the host. You know, and the host is an integral and an important part of these communication channels, and as such, it contributes to the perturbances that lead to microbiome-associated diseases, and we anticipate that the host could also generate interesting therapeutic targets if we understand the mechanism. So just to give you a brief example of a recent work in which we discovered the joint contribution of the host and its microbiome towards a very defined disease process. In this work, we we're interested in a very common, unfortunate human behavior, cigarette smoking, which, as you know, still impacts large proportions of our population, mainly in the developing world, and is responsible for many cancer and non-cancer related death. And in this project, we were interested not in the cancer part, which is equally interesting to us, but actually in the very most common reason leading to people either refusing to quit smoking or failing to quit smoking, which is an amazingly a large weight gain that accompanies smoking cessation. So we're talking about around 10 kilograms of weight gain within 1 year after smoking cessation. And interestingly enough, when people studied this phenomenon, they found that people who stop smoking and develop this dramatic weight gain do not eat more in most studies. So why do people gain more weight? So given our interest in metabolism and the microbiome, we asked ourselves whether the microbiome can contribute to this interesting form of obesity. So to study these three very talented people in the lab, Leviel Fleur, Uriah Moore, and Hagit Shapiro, it began, as we always do, with a very naive question: does the microbiome impact smoking cessation-induced weight gain? And they started by developing a simple mouse model for smoking and smoking cessation based on the placement of mice in a smoking chamber in a very defined condition mimicking intermediate level of chronic smoking. And they included groups of mice that were depleted for the microbiome by wide-spectrum antibiotics. And just to give you an example of many different studies that were part of this project, you can see here the red group, which are mice that started by actively being exposed to cigarette smoke in the left part of the graph. And you can see that active smoking results in weight loss, which is a very well-known and microbiome-independent phenomenon known for decades. And once we stopped their exposure to smoke in the middle of the graph, they developed this dramatic weight gain, which is what we also notice in humans. But what was most important to us and was interesting was the yellow group, which are identical mice that were smoking the same exact cigarettes. But once they stopped smoking under wide-spectrum antibiotic conditions, they developed a much attenuated weight gain following smoking cessation. And this tendency was very persistent. And even after one smoking cessation round, the yellow group of mice lost or gained less weight for many months afterwards. We thought at the beginning that maybe this was something related to our facility or to the diet that these mice were given. So we repeated these experiments with many different diets, with mice obtained from a different vendor and carrying a different microbiome. And even with low research nicotine cigarettes, very expensive cigarettes, that kind of were part of the effort to rule out nicotine as the simple explanation for this whole thing. And in all of these other contexts, the depletion of the microbiome by wide-spectrum antibiotics was associated with a quite marked attenuation of smoking cessation-associated weight gain. So the next question is, if the smoking and the microbiome are interconnected, then being simple people, we think that smoking must impact the gut microbiome. So we characterized smoking mice very extensively. And indeed, we found that both composition and function of the microbiome changed markedly in the smoking period in mice, which means that some molecules in the smoking released into systemic circulation must reach the microbes and do something in these microbes. And to prove this, we exemplified this by measuring nicotine in its main breakdown product, quaternion. And indeed, we found that in addition to the high levels experienced during active smoking of these exemplary smoking-related compounds in systemic circulation, they influx into the gut and could be measured in equally high levels in the gut of smoking mice. And if we took mice that never smoked but supplemented them with nicotine, we could see that the microbiome alterations were not identical to those of active smoking, but were in the same direction. Of course, we're not saying that nicotine is the major and the only cigarette smoke-associated compound that changes the microbiome, but this is just an example showing that these compounds reach the gut and they interact with microbes, change their activity and their composition. But as I told you repeatedly, we really find an importance to going beyond association and correlation into causation. So the next question we ask, does the microbiome altered, or does the smoking altered microbiome actually drive smoking cessation weight gain? And to try and prove this, we performed a very large number of transfer experiments into germ-free mice, the mice I've mentioned at the beginning of my talk, for mice that were smoking either for a short period of time or mice that were smoking for a long period of time, and mice after ceasing smoking. When we transferred the microbiomes from mice that were only smoking for a brief duration into germ-free mice, and all of the recipient mice, just to be clear, have never seen smoke in their life. They were not exposed to smoke whatsoever. We found that the microbiome was already changed by this short duration of smoke exposure, but it was not enough to change the weight of recipient mice as compared to mice that received a non-smoking microbiome configuration. However, when mice were smoking for a long enough period, the transfer of a microbiome induced a substantial weight gain as compared to mice receiving a non-smoking microbiome. And the biggest effects were noted upon transfer of microbiomes from the smoking cessation period, in which under many different dietary and other conditions, recipient mice develop a marked obesity, suggesting that somehow this microbiome stores an information that contributes to smoking cessation weight gain. Of course, the next question is one which involves a mechanism. What is the molecular mechanism of this effect? And as I told you, we are great believers in metabolites, and so we've performed an extensive mass spec-based characterization of the small molecule profiles along all the interventions that I've shown you before in trying to reach differentially abundant metabolites of interest. We've used sophisticated multivariate mixed models, and we reached 7 small molecules that kind of behaved in all the different conditions in a way that made them suspect as the drivers. And I'll focus on 2 of them, which became the ones that were validated. And these are 2 interesting small molecules. One is called D-methylglycine, the other is called acetylglycine, and they're both part of a very well characterized pathway which starts at dietary choline and involves both the gut microbiome and the host. And the first enzymatic steps in this pathway are generated exclusively by the gut microbiome, leading to the degradation of choline to betaine. And then the gut microbiome cannot further degrade betaine, and betaine is influxing into the portal circulation and into the liver, in which the host, through a very well characterized enzyme, converts it into bioactive D-methylglycine or DMG. We also know that DMG somehow generates ACG. This pathway is not well characterized, and we know that the levels of these 2 metabolites are a mirror image of each other. So when DMG levels are high, ACG are low, and vice versa, meaning that there is some cross-regulation which is not well characterized. And what we found, and I'm showing you just one example, is that an abundance of DMG and a depletion of ACG were strongly associated with smoking cessation weight gain. So in one of the experiments, we've tagged by isotopes the substrate choline sulfate and gave it in diet, and then measured each and every one of the pathway intermediates in the pathway I've shown you under the smoking and antibiotic depletion conditions. You can see that upon the induction of active smoking, the intermediates in this pathway are induced. So smoking induces this pathway. And you can see that when we give broad spectrum antibiotics, the pathway is depleted and the differences disappear, meaning that the microbiome in the first steps and the host in the last step, leading to bioactive DMG, cooperate during smoking in inducing the system and its products. So this is, again, what I've shown you for DMG going up during smoking and down during antibiotic treatment. And for ACG, for the second metabolite, this is a mirror image. You can see in red that upon active smoking, ACG levels are reduced. You give antibiotics, they go back to their baseline levels. Now, this is a very interesting association for us, but again, it doesn't prove causality. So to prove causality, we ask the next question: do DMG and ACG actually contribute to smoking cessation-induced weight gain? And to do this, we supplemented each of them in contexts in which they were depleted. So for example, for DMG, I've shown you before that when we perform this experiment with smoking and smoking cessation and deplete the microbiome by wide spectrum antibiotic, the weight gain is attenuated and DMG levels are also lower. So now we perform this experiment under antibiotic conditions, and we supplement the DMG levels that are now low back to their physiological levels, and we can fully restore smoking cessation-associated weight gain. For ACG, we do the opposite. We induce smoking cessation weight gain without antibiotics, and in this case, DMG levels are high, ACG levels are low, and now we supplement mice with ACG, and we can attenuate smoking cessation weight gain. So this suggests that high levels of DMG and low levels of ACG both contribute towards this weight gain phenotype. Another way to do this is to play with the dietary substrate. You can give mice a choline deficient diet. You would expect, and this is indeed the case, that the pathway would be depleted. And now when you smoke them and you cause smoking cessation, despite the fact that everything is in place, in the absence of the substrate and the pathway, smoking cessation-associated weight gain fails to develop. Now, we next asked ourselves a very curious question. We said, okay, we've discovered this interesting mechanism in the context of a very specific type of obesity, in the context of smoking. But once we've discovered these metabolites, maybe their use or their efficacy could be generalized beyond the smoking cessation context. So we ask the next question, do DMG and ACG actually induce metabolic effect even in the absence of smoking? So we took mice that are never exposed to smoke. We induce obesity by feeding them with a high-fat, high-sucrose diet. And when we gave them DMG, we could induce a mild but consistent weight gain. When we did the same and supplemented them with ACG, we saw a quite substantial tendency towards weight reduction. So this tells us that even if we study microbiome effects in a very narrow kind of context, in some cases at least, the downstream molecular mechanism could be generalizable. And finally, we wanted to see whether some of these concepts apply to humans. We've assembled a cohort of actively smoking and non-smoking human individuals. We could show by sequencing that their microbiome was different, interesting, and we can even look into some of the intermediates in the pathway that I've shown you in mice, and to show that it is induced in humans in a similar direction to the one that I've shown you in mice. And of course, this leads to many open questions related to these metabolites and to others, in maybe in the future assisting in smoking cessation compliance. And just to summarize this story, what we think we've uncovered is a holobiont situation in which, in the absence of smoking, the microbiome is geared towards the production of low levels of DMG and high levels of ACG, contributing towards a healthy and low weight in non-smoking individuals. And now a person starts smoking, and two parallel phenomena occur. First, there are known metabolites released by cigarette smoke that are completely independent from the microbiome, which have a very strong anorexogenic effect. So people who actively smoke develop a very strong tendency towards weight reduction. But what we've discovered is that in parallel to this weight reduction, the microbiome adapts by changing its configuration to one which favors a high level of DMG production and a much lower level of ACG production, theoretically at least, in an attempt to reverse this weight loss. Now, during active smoking, the non-microbiome related mechanisms are dominant and people don't gain weight. But what happens when a person ceases smoking, the anorexogenic weight reducing smoking related metabolites are out of the system within hours, they dissipate. But the microbiome is highly persistent, and we've shown this in many other contexts, and it continues to secrete DMG and to not secrete ACG, leading or contributing towards a tendency of weight gain following cessation of smoking. And we've also shown that potentially, at least by doing so, we may be able to generalize these findings beyond the smoking context. And just to summarize the talk, I think that one could cautiously say that the field is slowly maturing towards the study of mechanism and causality, and we hope that within the next decade, we would be able to gradually introduce a microbiome toolbox that would enable to integrate these and many other modalities of treatment into the clinical shelf, including in cancer. As you've heard, I had a cancer division. Many of these are being studied in cancer, but I chose kind of to ignore them today. So I'll finish by thanking the many people at my Weizmann Institute lab coming from all over the world and participating in this journey. our collaborators, our funding agencies, and for the past couple of years, our second hub at Heidelberg, the Microbiome and Cancer Division, hosting an equally great group of young researchers. And there we try to apply all the know-how and technologies that we've developed as the Weizmann in order to study whether and how the microbiome may impact cancer development, progression, and response to treatment. And with that, I think I'll stop here and I'll be happy to try and answer some questions. Thanks.
Thank you, Ron. It's, as always, I mean, you cover a lot of ground. I think it's quite overwhelming. Okay, well, we'll get started with Q&A. If anyone have questions, just raise your hand or you put your question in the chat box. I can just call your name. Well, I guess I'm gonna kick off. Well, I mean, there's too many things you talk about. I don't even know where to start. So I think I'm gonna start to ask about this vaginal bacterial transplantation. And I'm actually curious that, you know, with the fecal transplantation, we always know there's a interaction between the host and the microbiome. When you actually give the donor microbiome to the host, that's not only remodulate the microbiome community, but also shape the host genome in terms of the host, let's say, epithelial environment. Whether this is the same case with the vagina, I guess it should be a vagina host genetic microenvironment. Like, have you ever using the animal model or you can use I guess, an oncologist can use the epithelial sequencing to see after you transplant the microbiome, whether they change the genome, the host genome environment?
Yeah, I think that's a great question. We know very little about these interactions in comparison to the gut. We do know that there is a bilateral commensal interaction. For example, the lactobacilli in the vaginal microbiome, they love to eat glycogen, which is extensively generated by the epithelial cells in the vagina, not in any place else. And in return, they secrete factors that support the preservation and the growth of the epithelial cells. So for sure, there's data suggesting that such a commensalism exists. We know very little about what happens when bacterial vaginosis, for example, occurs, what goes wrong in a molecular level, and whether VMT, vaginal microbiome transplantation, restores this. So, you know, we have no answers to this. This was a clinically oriented first in human clinical trial. There's much to be learned in animal models and in larger scale human trials. I would say that if you want to draw some parallels to fecal microbiome transplantation, we increasingly are now learning that not all donors are created equal. So there are super donors, and we really don't understand in fecal microbiome transplantation, despite hundreds of thousands of procedures being performed, how to differentiate between the better and worse donors, even in the simplified scenario of a C. difficile infection. From our very limited information, which is now growing, it seems to be the same with vaginal microbiome transplantation. So not all donors in that context are also created equal, and there's much to be learned on trying to identify which are better than others and to kind of harness a donor to a recipient.
I see. The second thing I actually want to know is the model you're using in the LS. You always emphasize the causality of the microbiome and the host phenotype. But in this case, I'm actually curious what your comments on this scenario is. Do you think that the change of the dysbiosis somehow initiate the ALS disease, or is because the neuron degeneration or neuron dysregulation caused the dysbiosis?
Yeah, that's a great question, and I didn't go into the downstream kind of neuronal data that we've generated, which actually suggests that the microbiome doesn't cause ALS. This is for sure, at least not in this model and with this genetic perturbation, but it contributes to ALS towards its transcriptomic and epigenetic impact on subsets of neurons, and there's much to be learned there. So this is at least the suggested observational phenotypes are all out there. Interestingly, 6 months after we published this paper in Nature, came out another Nature paper in which another group studied the same exact question in a completely different ALS model called TDP43, which is a common sporadic ALS mutation. And there too, they found a very striking microbiome modulatory impact on that disease, but it was very different because that disease, in contrast to the SOD1 transgenic disease, was highly inflammatory. So there, neuroinflammation plays a much more central role in producing the neuronal damage. And they saw, again, in a different model, in a completely different set of suggested mechanisms, that products coming from the microbiome likely impact this inflammatory response in that model towards other directions. So what this teaches us is that there's likely more than one mechanism and more than one silver bullet metabolite that does what it does. It really depends on the context between genetic susceptibility, the type of microbiome, and the environment that you're exposed to.
But are you saying that the other model, which actually microbiome dysregulation, is somehow determination factor to control the development of the disease? That's what you're saying?
Even in that model, you need the TDP43 mutation in that mouse in order to develop the ALS symptoms. So it's not that you can take a microbiome in a wild-type mouse and develop ALS. So that would be a very gross and criminal overinterpretation. Yeah. But I think that the microbiome, through different mechanisms, could very likely explain the variability between people suffering from the same genetic predisposition, some of whom develop the disease and some of whom live happily ever after without ever developing it.
I see. But you actually think that the recalibration of the microbiome can somehow correct the neurological disorder?
Well, I don't think it, or at least our data doesn't suggest that it cures mice from the neurological disorder, but the delta, this delta symptomatic or phenotypic difference that we've shown, if true in human, could mean a few years of life or life quality or whatever. So I don't think that I would dare say that we can cure ALS. At least with the data we present.
But what about? I mean, I obviously recently there's so many different studies emerging in the microbiome related to the neurological disorders. I mean, give the simple example of autoimmune multiple sclerosis. That's also a quite. This is definitely an environment and then genetic combination for the disease, but it's different from the ALS because the ALS, I believe, it's more genetic dominant. The MS is more like a combination, but do you think that in that scenario, for example, in the autoimmune scenario, the microbiome probably have a more predominant function?
First of all, I wouldn't call ALS a predominantly genetic disorder. This is a very far from a monogenic disorder. It's a environmental driven plus genetic susceptibility disorder, just like obesity, IBD, or cancer, for that sake, in some cases at least. I do think that inflammation and its impact by host microbiome interaction is where we started when we started studying the microbiome just over 10 years ago. So there's much more, I think, detailed knowledge on mechanisms and pathways. So potentially MS, which has been shown or suggested to be impacted or associated with microbiome changes, could be modulated. But there also, I would be very careful not to overinterpret. And we really need to understand mechanisms.
Sure, yeah. The next question I have is about this. I'm really actually impressed with the Phages treatment. The cocktail which you actually utilize to treat the IBD patient. I'm actually, can you elaborate a little bit more detail that how you do the design strategy to actually which specific strains you actually design at the beginning in order to treat IBD?
Yeah, so basically, I'm not going to overly go into detail. This is unpublished data. 3 postdocs are going to hunt me afterwards. But basically, the process that we've developed is a very rational one, in which you start with a large, geographically diverse human cohort and very deeply try to characterize the clade of strains that cause whatever phenotype you're interested in. And the disease here is just an example for us. And then the interesting part, I think, is how do you come up with a phage cocktail?
Yeah.
And basically, the way we did it is a very painful way by generating many phages from lots of sewer and other samples, which target the family of bacteria that we're interested in, and then applied against strains that we've isolated in an iterative process in which we look for activity against this clade, and then many different combinations are used in order to see whether resistance stops being developed. So if you, again, if you put one phage on one bacteria, there's likely within the million or so copies of the bacteria in the culture, a bacteria that has a mutation in each of the genes in the bacteria. So one bacteria would be mutated for the receptor for the phage, and resistance is 100% likely to occur. So, but if you apply 2 phages or 3 phages or 5 phages, eventually in a very painful in vitro, ex vivo, and then in vivo process, you can come up with combinations that would not enable the bacteria to develop resistant phenotypes. And this is exactly what we did in this case.
Oh, I see. Okay, that's okay. I see. That's quite a lot of work. I saw that Yuxiang has a question. Go ahead, Yuxiang.
Well, I just want to thank you for this such a fascinating lectures, and I enjoy every single slide you presented. My group is working on immunometabolism and inflammation, and so the microbiome is really new area we're trying to learn. So I would really appreciate if you kindly comment on that with the current knowledge in the field. Is there a correlation between the microbiome profile with inflammation?
Yeah, it's a very good question. Many people have tested, mainly in humans, but also in animal models, the potential associations and correlations between microbiome changes that characterize aging and the aging process, even healthy aging, with both healthy inflammaging or non-healthy age-associated disorders, even Alzheimer's and others. And have found interesting associations. The problem is that these are associations and not causation. So just to give you an example, wonderful studies have shown that people that dwell in nursery homes feature a different microbiome as compared to those who live at home, at the community. You can even model the chances of developing age-related disorders based on the microbiome. But the problem is that these are very much confounded by dietary changes and other ambulation and lifestyle modification changes that characterize the different subpopulations of aging individuals. So it seems promising. The jury is still out there. With respect specifically to inflammaging, or TNF-alpha is a hallmark cytokine, the only one. Many people are suggesting that such correlations exist, but again, I would be very careful to deduce from associations and correlations to causations, because in most cases, the microbiome changes that are seen, especially in the human setting, are in fact secondary to disease and environmental impacts and not driving them. So the billion-dollar question is, how do you distinguish between the minority of microbiome commensals and their functions that contribute to a process, to the many that are secondary to a process? This is probably the biggest challenge this young field is facing.
I see. I saw some beautiful causation studies, and I imagine probably proof that point in aging probably is much more challenging.
Yeah, very, very long and expensive studies, I'm assuming.
Thank you very much.
Well, I mean, I have one more question actually related to the smoking study. I mean, obviously the choline actually treatment or choline influence have a big difference from the male-female. I mean, how you actually can comment on that, Eran?
Yes, so it's a great question, and in general, if we take it even one step further, gender differences in microbiome configurations, microbiome impacts, microbiome regulation by even sex hormones are ones that people have suggested and have shown in quite elegant studies to be present. What they mean, I don't know. In this particular study, we've interchangeably used the males and females. I don't have enough knowledge or information to directly compare between the two and to tell you whether, it's not that it exists in one gender and not the other, but If there are more subtle changes, we haven't gone deep into them. But in general, I think that more and more people are looking into gender-specific differences in the gut microbiome, for example, leading to gender-specific susceptibilities or contributions to different phenotypes. We're less of that part of, we're doing less of that part of work.
I see, okay. Well, I mean, if anyone else has question. All right, so if there's no other question, I'm sure if you have question, please just email Eran. I think he'd be happy. Oh, you have question?
Hey, hi, Eran. Haven't talked to you for quite a long time. Yeah.
Yes.
Wonderful scientific findings and the attempts to modulate nutrition or microbiota to intervene diseases looks quite promising. So I have only a general question for you. In industry, there are many microbiome startups such as Series, Avlo, Vedanta, Day2, BioX, and actually you are a consultant for Day2 and BioX, right? So these
I'm the founder of them.
They use different approaches to modulate microbiota to treat diseases. So in your opinion, which method might be the final answer?
It's a good question, and the answer is, I can ask the same question of which of the medical treatment for congestive heart failure is the final answer, and the answer, as we know it 30 years later, is that we in most cases need a combination of beta blockers and other modalities and ACE inhibitors. So our assumption is that the more of these efforts are underway and the more of these tools are put on the medical shelf, the clinicians will integrate them into medical practice based on evidence-based medicine. And I don't know what would work best in which combination. We follow what's interesting to us and what generates a signal. Probably, given the complexity of the system, I'm assuming, and from knowing, in almost any other multifactorial diseases, effective treatment would necessitate integration of different modalities together.
Yeah. Do you think the effect of a single bacteria or a bacterial combination would mimic the effect of like FMT? Do you think?
Yeah, it's a very good question, especially given the fact that I think Vedanta just published in Cell Host-microbiome today their phase 1 trial. I would say that it's very context dependent. If we take the bacterial vaginosis as a case study, probiotic therapies, including ones given the lactobacilli, which are dominant in this simplified microbiome, have failed. Well, we show in the transfer of an entire community that the effects are quite remarkable. And this is just an example of many other examples of the same kind, including in FMT. What that tells us probably is that a single bacteria lacks a lot of the support network that would enable it to take over the niche and to maintain itself over the prevailing disease-associated microbiome configuration. And one would need what I would call a core microbiome transfer in order to do the trick. So I think if you allow me to speculate, probably FMT would not be replaced by a single bacteria, but I am hopeful and I actually believe that the many people doing FMT research would be able to narrow down from this very blind whole microbiome configuration transfer into a defined consortium of microbes that would support each other and their colonization, because it's all about colonization and colonization resistance. And that would allow, on the one hand, to simplify the process to make it more reproducible. On the other hand, it would allow these bacteria to support each other. That's my guess. It's a speculation.
Mm-hmm. Yeah. Thank you again for the great talk and answers.
So I would like to ask you one more question. You early in your lectures, you show the beautiful data. You show the precision nutrition intervention affecting glycemic regulation. That was just so really impressive. And as you know, NIH have this new initiative about precision nutrition. As us in the nutrition department, we're really trying to leverage that. But do you think, you're the giant in the field. I'd like to pick your brain to see that, in your knowledge, do you see a good correlation between those microbiome profile versus this serum kind of, all this kind of metabolic readout?
Yeah, it's a fantastic question. We have looked into it and actually published on it, and there is a correlation. What it means, I don't know. 50 to 80% of all small molecules you would find in a healthy peripheral blood are associated in one way or another with the gut microbiome. So the association is clearly out there. But in the context of prediction and glycemic responses, and of course in mechanism, in this project, in contrast to everything else I've tried to show you, there is no mechanism, right? This is a black box. We've used machine learning to bypass this entire black box. The black box could likely include small molecule signaling. We don't know what they are. So I think that the association is likely to be there. The mechanism is also likely to be there, but we know very little on both.
So I guess it's probably the microbiome sort of have the potential to be a biomarker, but it's really need a lot of work to really, verify that's indeed the case.
I couldn't agree more. And if we were to do this in the conventional way without using AI, we would probably be looking at 30 more years of conventional mechanistic research without getting to the ability to integrate the data into an actionable result. So there are pluses and minuses. Here in this particular project, we were able to suggest that we can get to the result without understanding anything about mechanism and drivers. And now we and many others are trying to work our way back to identify these drivers.
Thank you so much.
Okay, so I guess if that's all of the questions, thank you so much, Eran, for joining us for today's seminar. I think it's really, a lot of information we can digest, but really appreciate your participation. Hopefully I see you soon in US, take care.
Bye guys, have a good one.
Thank you very much. That's fascinating.
Bye-bye.
Bye-bye.